Hello, my name is Michael Abacassis, and I am the Dean of the University of Arizona College of Medicine in Tucson. I am a professor of surgery and immunobiology. I am pleased to welcome you today to the 6th webinar of the Wonder at Home series entitled COVID-19 Antibody Testing and Immune System Support. The panel discussion will be moderated by Dr. Karen Lutrick, Assistant Professor of Family and Community Medicine. Our panelists include Dr. Janko Nikolich-Žugich, Bowman Professor and Department Head of Immunobiology, Co-Director of the Arizona Center on Aging, and the Principal Investigator on Antibody Testing for the state of Arizona. We'll also hear from Dr. Michael Johnson, Assistant Professor of Immunobiology and member of the Bio5 Institute. The University of Arizona's College of Medicine in Tucson has been actively involved in the fight against COVID-19, and I am excited that you will have an opportunity to hear from our faculty as they share details about their work with you this afternoon. As the new Dean of the College of Medicine, I am extremely proud of our faculty's engagement, commitment, and dedication to achieving excellence in our tripartite mission of medical education, research, and clinical care. I hope you concur. I will now turn it over to Dr. Lutrick, and I look forward to a stimulating discussion.
Hi, good afternoon, everyone. As Dr. Abacassis mentioned, I'm Dr. Karen Lutrick, an assistant professor in family and community medicine. Part of my research focuses on disaster response research, specifically connecting clinical and population research. I'm also a co-investigator on the Arizona Heroes Project, which is a CDC-funded COVID-19 surveillance project with Dr. Janko Nikolich as well, that is focused on understanding infection and immunity in healthcare workers, first responders, and essential workers that are on the front lines of COVID. We'll begin today with brief introductions by our panelists, followed by an opportunity for panelists to answer the questions that you submitted at registration. Throughout the discussion, you will also be able to submit questions using the Q&A function in Zoom. I will review and ask panelists to answer as many questions as we can. We'll then wrap up with any final thoughts from our panelists. So first, I'd like to start by inviting Dr. Janko Nikolich, an internationally recognized leader in immunology and aging and the PI of the Arizona Antibody Testing Initiative, to start with some opening remarks.
Thank you, Karen. It's a pleasure to be here. So just wanted to reiterate some of what you have mentioned. My laboratory is really interested in how the immune system operates and how it defends us against microbial pathogens, viruses, bacteria, and so forth, and also how that capacity wanes with aging and what can we do to fix that. On a side, we're also interested in generally trying to improve not only the length of life, but really the quality of life in older adults. So those are some of the things that we think about all the time. And then when there's a virus like this that affects older adults disproportionately, then we typically run and study it.
Thank you very much. And now I would like some opening remarks from Dr. Michael Johnson, an assistant professor in immunobiology and the Bio5 Institute.
Hello, everyone. My name is Michael Johnson. I'm an assistant professor in, as she said, immunobiology. Janko is the chair of the department. My laboratory studies actually bacteria, but moreover, we actually study how metals impact biology. So if you look at the bottom of your vitamins, you know, those things right at the end, things like iron or manganese or zinc, those are some of the things that I study. It turns out about 40% of all your proteins use metal, which has a big implications on how it affects bacteria, which is what I primarily study. But also we're looking at how some of these metals might impact for certain viruses, specifically SARS-CoV-2. And we also are interested in seeing if we can take advantage of some of these metals and how they affect those particular viruses by finding different compounds that work in concert with those particular metals to maybe break a certain protein in a very controlled manner. So that's what my laboratory has kind of shifted focus and is starting to work on as of currently.
Thank you. Maybe we can start with sort of a broader question. So from both of your perspectives, how is the university beneficial in moving the science forward and sort of combating this COVID-19 pandemic? Janko, would you like to start?
Sure. So, university unites people in very diverse scientific disciplines, as well as people that deal with clinical care, people that deal with population aspects, spreading of the virus and so forth. And that is really the power of having the colleagues that think about all of these different aspects that you don't think about usually, and then providing whatever your expertise is to that, whole that is much bigger than the sum of its parts. If I weren't at the university, I would have no way to access patient samples that we can get from the hospital. This was a virus that we knew relatively little about. In some ways, we knew a lot about its nearest cousins, but not about this one in particular. And so studying it without being able to get access to the materials early is virtually impossible. Also, when we're thinking about what we have put together, which is an antibody assay to test for antibodies and immunity in people, that would have been completely impossible outside of the setting of a large university. And not only that, but with everybody really wanting to do the right thing and wanting to help and wanting to forward our mission so that we can help our community. So we had a robotic facility that was already certified by some of the highest certification levels that you can have from the FDA. That facility was essentially repurposed for not only the testing of the virus itself by this PCR method where you get the nasal swab, but also for our antibody test. And, so there's many examples where, we would not even be at the beginning, much less where we're now, in terms of being able to understand and rapidly study the virus and help in turn with hopefully interventions and actionable information that we can use to help the communities.
So I think it's been very interesting. I kind of started off by saying that I'm a bacteriologist, so I deal with, some of those other tiny living things, and definitely not a virologist by training, although we have some very good ones at our institution, which has been great because now it means that we get to be better than the sum of our parts individually and come together. So I would not have been able to join the fight against COVID if not for my colleagues. But inspiration, I think, can come from a lot of different places. And I'm just very fortunate to be able to be a part of a university that cares about teams and team research and collaborations, because, when we're dealing with more and more complex problems as a global society, we have to really come together more often to kind of make these kind of groupings to move that particular science forward.
Thank you. That's really helpful. So in general, you talked a little bit about this, but really specifically, Janko, it seems like your research has really changed and had to pivot because of the pandemic. How do you see that continuing to change, and how has that changed your vision for the future of your research?
Well, it's not the first time that it has pivoted. When we had the West Nile virus outbreak, that was the first time that I kind of as we would say, chased the bug. It was something that we haven't worked with up until that time, but it is a virus, and we do a lot of viral immunology. We try to study the immune response to viruses. A lot of it, I think, depends on how quickly can you get the reagents, how quickly can you get the samples so that you can ask these meaningful questions. And in this situation, I can tell you that for the first 2-3 weeks, I was a little reluctant, maybe even longer, maybe like 4 or 5 weeks, starting from late January, where it became clear that this thing is spreading and is likely to affect large swaths of the world. And I was thinking, well, obviously it started in China. They're going to have such a big head start. It was spreading through Europe. Everybody's going to be in line before us to be able to even, start studying all of this. And also the initial signs from what I read about, the virus and what I knew about SARS-1, suggesting that, I really like to study. The immune system has 2 big lines of defense. One is called the innate immunity, and it's highly conserved all the way down to fruit flies and worms, and to us. And that's like the primary and early and somewhat primitive but very efficient way to alert the body that there's an infection. After that, our organism in all of the vertebrates has developed a second layer called the adaptive immunity, which consists of white blood cells and very specialized white blood cells, T and B lymphocytes, some of which make antibodies like B cells, some of which attack the virus more directly like the T cells. And I care about that latter part of the immune system a little more. It's closer to my heart. That's how I was trained. It looked originally that a lot of the fight with this virus may be determined, and a lot of pathogenesis, bad things that it does to the body may be related to the innate immune system. So I was like, kind of on the fence for about 4 or 5 weeks. And then it was, no, this thing is coming to us. We all need to study it. There's no help really coordinated coming at the national level. Testing is slow. We need to put our own tests. We need to take care of our own population. Let's do it. So that pivot was there. How will it continue? I mean, I see this, and that was the other part, I think, while you decide whether you can jump into something transiently. But, is this going to be for a long haul? I think, yes. This group of viruses has jumped into the human population in less than 20 years 3 times. And each of the 3 times we got pretty nasty infections. SARS-1, 8,000 people sick and 10% dead. MERS, coronavirus in the Middle East, about 30,000 people, a little less, but about 25% death rates. So, and then this one, which, is less deadly, but it spreads like a wildfire. So these viruses are going to be around to stay. And I think all 3 of them were attacking the older population, the vulnerable populations, they're not going to go away. These events are going to continue. We need to be prepared as both the academic institution and as a society for this. So I think I'm going to be in this for a longer haul. And then it becomes, the question becomes, how do you build the infrastructure so that we're far better prepared for the next onslaught relative to where we were with this one? And that might be another question for discussion.
Yeah, I'd like to come back to that because infrastructure is something that I think a lot about. But Michael, do you want to share kind of how this has changed your work as well?
Sure. I wasn't actually going to get involved. My laboratory was shut down for the safety of everyone involved. And then I kept getting people saying, hey, have you heard this research about the virus in copper? The virus in copper. And copper is actually the metal that my laboratory deals with the most. And we had some treatments for, some kind of, like I said, some synergistic treatments or ions that match with copper that could actually, or chelators that match with copper that could actually kill bacteria a lot faster than just copper alone or the compound alone. So we were actually interested in maybe, if it works for bacteria, if it works for things like cocci, then maybe it can work for a virus as well. So we were that kind of brought me into the fight. And, it's interesting. I'm an assistant professor. I'm still trying to really establish my laboratory and my name into the community for the research as far as doing metallobiology, which is what we call it with bacteria. But, if I got into this field to really help people's health and to help protect global health and do what I can to find cures, then this would be the greater good of that. And sometimes you have to make sacrifices just for the greater good. So that's what I've at least tried to do. And I'm happy to not only have the support from the institution, but also the support from my department as well, because it's, it makes it easier to get into that fight. It gives you more tools to get into that fight. You're not just there, throwing a soft jab. You get in there with the team being able to throw an uppercut.
Yeah, thank you for that. And so I just want to sort of add from a more of a disaster research framework. A year ago we had the NIH out at the University of Arizona, and we did a workshop on a train derailment, right? But one of the the biggest things that we focus on when we're thinking about research, response and research, right? So responding to a disaster like a pandemic or a natural disaster, and then researching it is collaboration and integration of data, which is an infrastructure issue, which is an operations issue. And I think we often take for granted that if something like this happens, that the floodgates open and there are no more silos and we can easily share data and patients and collaborate and those sorts of things. But those things don't go away. And so removing those quickly and setting up infrastructure where we can really quickly get up to speed, connect studies ethically, responsibly with the community and the IRB and a variety of researchers, or we have clinicians and immunologists, and then also population researchers working collaboratively and sharing data and sharing ideas is something that doesn't happen easily. And we're grateful that we were somewhat prepared in the sense that we sort of started that infrastructure and we were able to fairly immediately test it and then expand it by about 100-fold so that we can do a lot more collaboration really quickly. And again, just preserving resources and time, because time is a huge variable in these sorts of things.
And I just like to add something here. I'm glad that you said all of this, Karen, because while Michael and I are allegedly experts that are being interviewed here, Karen's no less of an expert, and having her in this role and having her do what she did to connect everybody and to do exactly this, to unify the infrastructure, to break the barriers and make things easier for everybody else, amongst us to do this work is incredibly precious. I can't even begin to describe how important that is, because otherwise we as you said, we stay in our silos. We can't actually realize our potential. So thank you for that.
My pleasure. It's exciting work. It's nice to get people to work together, sometimes for the first time, but we always think of it as like starting a collaboration, right? And it continues to build. So one last kind of general question, and then we have some really great questions that are coming in for our panelists. But so thinking about in a perfect world, right, where we had all of the resources that we needed, what kind of impact do you think we could make? So what are some of those critical pieces of infrastructure that we as researchers at the University of Arizona feel like we're missing that maybe the community can help with?
Wow, I mean, that's a great question, and it's not the one that I have deeply thought about, but it's always really how can you connect to people who deliver care? How can you, because that flow of, ideas and information, this is an example of a health system under crisis, as well as the country under crisis, overall. And, when you're in crisis, nobody has the time. But there is something that, strategically you need to think about of, what are the things that you absolutely have to make time for so that we can make sustained and important progress. And so, one of the big challenges is for me finding, my clinical partners. I mean, I'm not a practicing physician. I have my MD, but I'm not in practice. And so, again, I talked about this a little bit, getting samples from people that are sick is incredibly important. And I understand, of course, that my clinical colleagues are, up to the wazoo caring for people and also concerned about, their own health and safety and their colleagues' health and safety and their families' health and safety and everything else. So it's really like, they were at the breaking points, nurses, physicians, everybody in the healthcare, and I'm coming in, I'm telling them, look, guys, I really need like a little bit of blood from this person or a little bit of something else, like, that they can give us. And they go like, well, this is research, that's not important. Now, that is a legitimate reaction in some ways, but on the other hand, in this situation, we can't move on this virus without that help. And so that was one of the things that, we gained upon, and we're, going to be a whole lot better off going into the next situation similar to this, as well as, what you mentioned, Karen, like, sharing information it's not straightforward. I mean, these are people that are undergoing care. We're taking their samples. We need to make sure that privacy is protected. At the same time, once I get something that I de-identify, I want to share it with everybody that's, like virologists that need to figure this out, with Michael and other people who are looking into new drug design and so forth. So how do you do this quickly? How do you do this efficiently? Those are the challenges that again, we have met, I think, wonderfully well. You can always do better, and you can always build on what you did well and learn from what you didn't do quite well. So all of these, I think, are going to be the legacies that are going to stay with us. I mean, it's a defining event of a lifetime in many ways, and I hope we can retain the best and learn from what we didn't do so well.
So I'll actually go in the other direction. I mean, I think a lot of the focus, and rightfully so, is on COVID-19 research. But I would ask, what's going on with all of the other research? There's diabetes research, there's heart disease research, there's cancer research. And you have to also think that a lot of these programs, and we have a lot of those laboratories here at U of A, a lot of those programs have actually been sidelined due to COVID. And then you add another layer to that. You say, well, how do we now train the next generation of scientists and the next generation of medical doctors? Because some of the training that they were going to get this summer was, again, not available because they couldn't travel. So now we're creating kind of gaps in the ability to create knowledge, not necessarily knowledge itself, but the gap in the ability to create knowledge. So how do we create infrastructure to kind of replace that gap or to try and overcome that gap? And I'll take this time to share one of the programs and initiatives that I've done this summer is there's a lot of people who will be applying to graduate school in the future, and they haven't been able to actually do that summer research project that gets them over the hump to actually apply and have a good CV and a good record to apply. Well, how do you provide them remote opportunities? Well, I came up with a project called the National Summer Undergraduate Research Project that actually helped link people in a virtual space to do different summer research. And what's been great about that is we actually matched over 250 undergraduates who are Black, Indigenous, and Hispanic to research opportunities. And some of those research opportunities have been dealing with microbes. Some of those have been dealing with COVID and actually modeling new drugs. And, we've been able to kind of keep this community going to help foster the next, at least try and keep the ground as fertile as we possibly can for the next generation of scientists.
Yeah, absolutely. And that's one of the things that, at least on the clinical side, where we're really trying to balance business as usual, taking care of patients, answering important questions that aren't COVID, as while also answering those important questions. And it's complicated.
And I think what you raised about the impact of this on chronic diseases and on people's health in the long term is looming large ones already and will loom even larger down the line, obviously. So
I'd like to transition into some participant questions. And the first one, and I think the one that's on a lot of people's minds, is the comment on the evidence that COVID-19 antibodies look like they disappear after a few months, and what that means for vaccine development and deployment. And so I know this is a favorite question of yours, Janko, so I think we'll start with you.
Yeah, it really is. And it's been unfortunately one of the misconceptions that has been perpetrated a lot, and it's really absolutely not true that antibody immunity. We've measured antibody immunity a lot, and we know quite a bit about antibody immunity, and it's and we're starting to study T-cell immunity, which is also going to be very important, and it's on right. There is no way that a virus like this would not create sustained immunity. We know that we mobilize a lot of resources when attacked by a virus of this type, and antibodies come kind of like last on the scene in some ways. I mean, they really do not show up. They're not very detectable until at least a week. And if you look at the cohorts, and we've just sent a pretty important paper out to review this morning, where we have analyzed more than 6,000 people and so forth. In our hands, immunity is really stable. But there is an explanation actually for why some people have concluded that it may not be. There's actually two explanations. One is when we looked at different parts of the virus, and we've looked at three different components of the virus for antibodies. We also looked at the ability of our antibodies to neutralize the virus, which is really, really important. It's a tough assay to do, so you cannot really run it on the whole population, but you can run it on on hundreds of people probably and so forth. When you put the virus together with antibodies and you put them on some cells that the virus would normally infect, if the antibodies are neutralizing, there will be no infection. We normally see the infection by the virus killing the cells and leaving these little holes on a layer of cells. So there's these pox or plaques, as we call them, are there normally when the virus is active. When the virus is disabled by antibodies, there's no holes. And that, by that measure, at least about 4 months out for a subset of people that we have studied, the immunity is absolutely stable. There is now evidence going back to SARS-1 that for people who got sick with that virus, this neutralizing antibody immunity is stable for at least 12 to 17 years. It was discussed that it was much less again, but those were different types of assays. So what we did find out is that antibodies to some parts of the virus do go away relatively quickly. But the ones that are probably for the more important parts of the virus are very, very stable. So there is some heterogeneity. So if some groups measured antibodies for one part of the virus, they could see them really drop down because they're inherently going to be less stable, although even that in our hands didn't really run to zero. The other explanation for this is that, we have this immunity that really ramps up. So antibodies go sky high. And at the point when the virus is controlled, they start declining. You don't need as many of them. And so if people start measuring them in that decline phase, they can extrapolate that, if it continued in that downward trend, it would really run into the ground. There will be none at all. The point, though, is that we have two types of antibody-making cells, and one of them is responsible for this big spike, and they're relatively short-lived. But as they are going down, there is a much more stable wave of antibody-producing B cells that tend to be around for years to decades. They pick up, and so typically as this one is going down, there's actually flattening, and that flattening, or maybe then slow decline, is around for many, many years. And so if people have just looked at this initial decline, said in these 2 weeks it dropped this much, so obviously in the next 3 weeks it's going to be gone completely. Well, you have to measure that. That's a wrong extrapolation, because biologically we know that this is not what happens and this is not what makes sense. So every antibody response has that decline phase. Even the most stable and the strongest ones show it. So in our opinion, this is not going to be a disappearing immunity. One question remains for people who got perhaps a very low dose of the virus, who were without symptoms and so forth, whether their antibody responses are going to be similarly long-lived as those that had a wallop of the virus and had to make a lot of antibodies. That we don't know for a fact yet. We had a very small cohort, probably about 6 or 7 people of that type that we have analyzed in our study that we just sent for publication. But even in them, we've seen those antibodies around for months at least. So, some of it is to be continued, and you have to persistently measure immunity, but I am not worried that we're not going to have immunity to this virus. And that's consistent with, how this particular type of virus is behaving.
I think one thing with that is, let's say you're measuring antibodies at their peak as far as in a person, and you're looking at the cells that make those things. Well, let's take a step back and kind of think about what happens when you are sick and when you are making some of those B cells and T cells to try and fight off whatever virus or bacteria you might have. You expend a lot of energy because you have to make lots of cells. There's a lot of metabolism. There's a lot of energy. There's the energy consumption for your body in that particular point in time is enormous, which is part of the reason why you kind of feel tired and achy. Imagine having that particular exhaustion for every single virus that you encounter that might be bad. And it basically would end up building on itself. And you would be more tired and more exhausted. And I know sometimes we feel like that is the case just based on how our day goes, but it is not sustainable. That level of actually, that level of, trying to keep up with, maintaining immunity against everything at that significantly high level does not work, which is why we actually have cells that can ramp up when they see the challenge instead of being ready for literally everything. Also, I don't think our bodies would be able to contain all those cells. I mean, we would be walking around as like Stay Puft Marshmallow people just because we would be full of all these cells ready to encounter everything as soon as, it can at maximum velocity. It's just not sustainable. And that's why our bodies have been, fine-tuned to be able to respond to things over time and, 10 years later, because we make these memory cells that can do that.
So, Michael, that's a great point. I'll just make a very quick comment in that's why you shift to these longer-lived cells. And there's many fewer of them, but they produce antibodies really efficiently. And there's been studies showing that only 3 cells of that type can actually confer protective immunity for a long time. So that for that very reason that you just said, we have to economize. We're there's 1 unit of each one of us and many units of different pathogens, and we have to be able to deal with that.
3 out of hundreds of trillions.
And so then translating that into vaccine development and vaccine deployment, right? There's a lot of questions about boosters and longevity and efficacy, and that's obviously tied to some of these sort of unknowns that we're still trying to figure out. But Janko, from your perspective, where are we in kind of starting to answer some of those questions?
I think one really important point to be made here, I made a case in what we know and saying that, I think immunity is going to be long-lived to this virus. But when you're making a vaccine, you're no longer bound to use the virus itself. So even if this virus, by any chance, would not give us sustained immunity, we have sufficient knowledge to engineer a vaccine to give us sustained immunity. And that's actually, that makes sense when you think about what is the job of a virus. The job of the virus is to multiply itself like crazy and avoid your immune response. So every virus known to us has what we call evasion mechanisms. They have evolved to try to avoid our immune system in different ways. When you're engineering a vaccine, you don't want to do that, of course, and you don't have to do that. You're going to take only the parts that are not, evading immunity, and you can use that isolated part together with other compounds that we know are going to awaken our immune response, to juice it up, to make a good immune response. So the response to the vaccine does not have to, equate the response to the virus at all, and there's no reason for it to not be better. Now, where are we right now? There's several preferred candidates that got a lot of money from the government in this Operation Warp Speed, and that was a great idea to infuse a lot of money into different types of vaccines. It's kind of like you're betting on multiple horses. These are not the only horses that are in the race, because other pharmaceutical companies that didn't get that money right away also decided this is a good idea. So we currently now have about 130 vaccines under development. Are we going to get a perfect vaccine right away? Maybe not, maybe yes. With 130 of them, we have, much better chance than with one. And again, you have to remember one thing about vaccines and vaccine marketing. They always go, like somebody mentioned in one of these debates, well, we've never made the vaccine against the coronavirus. That's true. We've never had a reason to make a vaccine against the coronavirus because, we have some that we don't care about because we handle them really well, the common cold ones. And, SARS and MERS, which are nasty, really didn't spread very much. So, no vaccine manufacturer is going to go and run and make a vaccine for 30,000 people. But, when they know that the market is now 20 million that are infected and a whole lot more fearing it, they have every reason economically and population-wise to go and make a vaccine. What is the length of immunity going to be from a vaccine? I mean, vaccines always have this balance between efficacy and safety. And it used to be that, all of the vaccines that were super efficient, like live attenuated yellow fever, wonderful vaccine, awesome, makes some people really sick. Smallpox vaccine, same thing. Not only that it makes some people sick, it can kill them, but when you have a disease that's killing 20% of the population, you take that risk. We know, as I said, a whole lot more about microbes now than we did 50 years ago, and we can make a whole lot better vaccines. So what comes in the first wave should hopefully protect broad swaths of population, particularly those that are most at risk. We will know pretty quickly how well they're working, in terms of both side effects and efficacy, and how long-lived this is going to be. And then we're going to take a second shot at it, and go and improve them. If we get a perfect one right away, great. If not, we're not going to stop. That's kind of like the long and the short of it. Seems that by what I'm, hearing, that we're still in the timeline of hopefully some serious, massive distribution of the first efficacious vaccines somewhere around December, January, February. We'll see.
Great. I just want, Michael, did you have something to add about vaccine?
Well, I just wanted to make a comment about what people are seeing as the scientific process in motion. And that is not always the most linear process. You see our successes, you see our failures, but we don't always see them as failures. A negative result doesn't necessarily is not necessarily a failure. It's, again, as a way to not make a light bulb, if you use that analogy. So I just want to caution people to say, well, if that didn't work, then how can I trust scientists? No, this is you're just seeing the scientific process in motion, and you're seeing it act out publicly as well, which is, like I said, it can be messy, but it works.
Yeah, it's a bit, bit frightening to see that, for people who are not used to that. But, we're used to a 90% and higher level rate of failure, because we are researching things that we don't know. We put together an idea, which we call a hypothesis. We try to test it, and we have to, the best thing you can do is test it as rigorously as possible. You're trying to make sure that you're right, but you're trying also to prove yourself wrong in the process. Because, if your idea in the end withstands all of that testing and is right, then you're going to contribute something meaningful. And again, as I said, you go into a big unknown, and we have plenty of big unknowns. You're likely to be wrong all the time, and we're used to it. And it's, as Michael said, a good negative result that tells you what's not true, really sets you to focus on the likely set of things that will be true, and that's super precious. Now, seeing, all of that in public, I'm sure is very unnerving. So that is where, we're trained to think like that and to operate like that. And, I want to prove myself wrong. And if I don't, then I might be right, and that's great.
Yeah, thanks. I agree. And I think there's also this timeliness, right? So studies are being done and published so much faster. And I know on the clinical side, so like Michael worked with some undergraduate students to get them research experience. I've been working with a group of medical students this summer. And one of the things that they're so concerned about is they're sort of preparing, they're at the end of their medical career. They're preparing to be physicians next year. Is like, how do you process all of this information if you're an emergency medicine physician? And there are just so many articles coming out every single day. How do you understand it, and how do you know how to treat that patient with the most up to date? And I think, we're also, it's sort of this second wave of like understanding how to digest research, understanding how to interpret what's a good study, what's maybe not a bad study, but like we're just not quite sure that their conclusion is what it should be, or we're not quite sure that that's reproducible. And so it kind of, they're missing out on some opportunities, I would say, because of the pandemic. But I think they're also kind of come out of this experience much better versed in kind of understanding emerging science, what translational research is, right? This whole bed to bedside, this whole kind of loop, and how it impacts them in a really exciting way that I'm not sure they would have gotten otherwise. So I'm an optimist, right? So I always look for the bright side. That's my bright side of spending this summer with medical students who couldn't do their normal clinical rotations. I want to transition though, and I know, Michael, you mentioned that you work in metals. And we have had a few questions who want to kind of from participants who want to understand the role of copper. We've been hearing about copper in masks, copper in cleanings, copper surfaces. And I'm wondering if you could sort of share, your experience. You're kind of our metal virus expert.
Well, metal maybe, but virus, I'll leave that one to Janko. But, there's a lot of, let me start off by saying this. I could probably be a millionaire right now if I basically endorse all of those products that people are asking me to say, well, I put copper in this. Is this now going to fight against COVID? But, ethics are a real thing, and I try to maintain them as much as possible. Although a nice Jaguar would be nice, right? But no, I digress. No, so how does copper work? Well, that's actually one of the things that my laboratory is trying to figure out. How does copper causes toxicity? And one of the things that you need to know is that copper actually works on contact. And we've been using copper since antiquity. So since, people used to have their food in copper pots to prevent their food from spoiling. They actually put it into various mixtures, like the Bordeaux mixture, which helped protect people's vineyards. So if you like wine, think copper. They sprayed the same thing on potatoes, and it helps stop the potato blight. So if you like French fries, think copper. But it still doesn't quite say how copper works. And we know that copper as a metal can actually come in and knock out some other metals that might be needed in that particular protein or complex. We know that copper can come in and, maybe cause some hydroxy, radicals that could actually help, as far as help the toxicity. But what we do know is that it works on contact. And if you have a mesh, a mesh that you can see holes through, then that's not really going to be an effective treatment against COVID-19 or any virus or any bacteria or any fungi or any parasite. What you really need is a actual surface, and not a coated surface. I'm talking about like the Statue of Liberty type of surface that will end up turning green kind of surface, that's why the Statue of Liberty is green. It actually is a nice brown copperish color if you get rid of some of that oxidation. So what I will say to that is be leery, be mindful of all those products that people say that this has copper and it will protect you against X. No, still wear a mask, still wash your hands, still wash your sponge, even if it has copper or silver in it. Still wash those copper socks or copper hand gloves that you might use to do weightlifting. Because quite honestly, one, there's a lot that we still don't know about how it causes this toxicity, which is, again, what my laboratory is trying to focus on. But there's also a lot of false claims out there. And I think that you can say, saying this is an FDA-approved compound doesn't mean that you're using it for an FDA-approved purpose. So I think people really need to be careful in, trying to evaluate some of those questions when they see them in an article, when they see them on a website, when they see it on a commercial, because I'm not going to expect you to dive into the scientific literature, although I can hopefully help explain some of those things to you. But I do really hope that, I guess, from things like this, that you get the perspective that you still need to have a critical eye when you read certain things, and just don't take everything at face value. If that article doesn't actually cite a publication or a literature, then maybe it's not the best thing. Try and dive. So I just want to encourage everyone to dive deeper, especially while we're trying to figure out all these treatments in copper in those particular realms.
Thank you for that. We received a handful of questions that are related to age. So it looks like, some ages are less affected by the virus, some ages are more affected by the virus, and it's presenting differently. And so I wonder if we could sort of just talk about how age is related to COVID-19.
I'd love to, Karen, but let me, there's four questions that I think I can very quickly answer that are going back to the duration of immunity and everything else. One is how do some people have pre-existing immunity that might protect them somewhat? Where there are coronaviruses that are in the circulation, they seem to infect about 96 or 98% of the population. So very few of us have not had an experience with a coronavirus. So some of us may have and likely have some protection from that. The second one was how is it possible that some people get COVID and get recovered and then they get it again? There is no documented case of that happening, actually. There's been anecdotal discussions about that, but we have not seen a single case where somebody really had the disease and came back with it. So that's right now a unicorn, and so we'll leave it at that. The third question was, would you expect that the vaccine will need a booster or will not need a booster? That will depend on the vaccine and on the strength and durability of immunity. So many vaccines need a second shot or a booster at some point. Very few do not. So it's probably more likely that we might need it at some point. So we'll see how that goes. That's kind of like in the future and hand waving. And then another question came up about the cytokine storm. If you have an aggressive immune system, we're not sure that this is just an aggressive immune system, but we don't understand why a subset of people really come up with this huge inflammatory response in their lungs. But there are treatments now that seem to be very effective at calming that down. Many are in phase 3 trials. I'm aware of one, for instance, in India with a compound called ulinistatin. There are some angiotensin related ones that actually deal with the molecule that the virus uses to get inside our cells. There are anti-cytokine antibodies that are also being used, particularly the one against interleukin-6. So all of that is now broadening our ability to treat these severe cases. We still haven't set up on one optimal treatment regimen, but they are out there and they're being used in the clinic. Going back to aging, and sorry for the digression. It's it's a fascinating topic. Why we age and how we age, what happens to our cells. It's one of those things that everybody seems to know when they see it. But there are, you can also look at the medical charts and you can compare two people and you'll go, wow, they seem to have identical medical history. And then you look at them and you go like, well, one person is not having really a great quality of life and the other one looks great. And it is the whole point really of the science of aging or which is called gerontology and geroscience more recently, is to make sure that we enjoy high quality of life as we grow older. So what can we do there and so forth. And the thought that is quite revolutionary and really interesting is that there are some processes that happen with aging that also happen in many of the chronic diseases that we suffer from. Like Michael mentioned, diabetes, metabolic diseases, cardiovascular diseases, neuroinflammatory diseases, and like Alzheimer's, for instance, and neurodegenerative diseases. All of them have certain things in common, and we think that there may be some ways in which we can treat one underlying cause and affect many diseases. That's kind of like the most exciting thing and the most exciting idea that exists now in the biology of aging. And so inflammation is one such thing. I just touched upon it. Inflammation is a super important, great acute process. You get attacked, you mount an inflammatory response, you get rid of the invaders, and you're done. Then you're supposed to switch off put the switch off and then have no longer any inflammation. Well, we know that as we grow older, that inflammation kind of like creeps up a little bit, and we very often have it around. And so regulating that inflammation may be beneficial in this case, both in terms of how we respond to an acute disease like SARS coronavirus 2, and for our general well-being. So those are the intersections that we really are interested in. How can we get that, how can we get that going and employ it in clinic, really? So, when it comes to the immune system, we have some fewer cells to fight the virus, and they're not as coordinated as they were when we were younger. They don't quite go as quickly to the site where they're supposed to go. And when they get the signals, they react a little slower. So there's a lot of these, tiny little defects that, if they're along the same pathway, in the end, you're going to end up with a big defect where the virus is going to outrun you. So the question is, how do we do that? How do we rejuvenate the immune system? How do we immunomodulate in the right situation to boost the right response and to turn off? One of the ways in which the viruses love to subvert our immune system is to make it think that it's been attacked by something different. And that's called immune subversion. So we know what kind of response we need to mount to effectively get rid of the virus. But if the virus, basically tells us, no, I'm a fungus, you start chasing against the fungus, that's not an effective immune response. So again, dissecting these signals and being able to modulate them as the infection occurs, and also within vaccines, is really the key trick. So, for instance, for the lungs, we know that, if you get neutrophils, which is one type of our white blood cells, they come in and they fight the bacterial infection or a viral infection, but they're hugely inflammatory. You're going to mount an enormous inflammation in the lung. And you want to regulate that. One of the cells that we have is also an immune cell. It's called the macrophage. It comes in and eats up, particularly the dying neutrophils, which are the most inflammatory of them all. And, we know that in older adults, these macrophages lose the ability to gobble up as efficiently and remove neutrophils to dampen down inflammation. So that's a good example where, dampening inflammation somehow is really important, and where the older immune system does something that is not quite as beneficial as you want. And so the more of these defects we isolate, the more we're able to intervene and do something about aging in particular situations.
Yeah, thanks for sharing that. And I will just add on the clinical side, one of the ways that we traditionally study things like pandemics, and it's kind of a model that we've done with influenza, is we really start with the critically ill. So we start with the folks that are in the hospital, and it takes us a little while to get to understanding the non-severe, right? So the folks that are just being treated urgent care, or maybe just treated at home. And so on the clinical side, we're really just now kind of shifting to caring not just about the people that are in the ICU, but the rest of the population. And I think that's why we don't know as much about how it impacts sort of across the ages, just because it takes us a while to get outside of the hospital. It takes us a while to get outside of ECMO and intubation, because it becomes these really important critical issues that are related to resources and infrastructure. And we're really now starting to understand who's coming into family medicine with COVID, right? And so we're collecting that data alongside, and we're hoping to maybe have a little bit of a better understanding of these non-severe. We're also not collecting as much blood from non-severe patients, right? Because they're harder to find, because they're at home and they're okay. They're sick, but they're okay. They're not seeking care. And so it's, again, another kind of integration between population. How do we find them in the community? We know where they are in the hospital, but how do we find them in the community and the urgent care, and start to really fully understand the full spectrum of disease?
Yeah, studying that resilience and successful responses is as important than studying the ones that are not so successful, and then we really know.
Yeah, and they're harder to find because they're living their lives. We're not seeing them every day in the ICU. So we only have a few more minutes, and I know we didn't answer everybody's questions, but we will be sharing as many answers and resources after as possible. But I did want to give Janko and Michael a minute to kind of wrap up and share any last thoughts as we end our time together today.
I'll start off by saying I have cousins who are MMA fighters who just got COVID. So in peak physical health and condition, but he just literally just got COVID. And one of the reasons why he's taking it a little bit more seriously is because he actually has, a scientist in the family to talk. So I will say that there's a lot of people who don't have access to that information. So I hope that you all are learning something from this, and that you spread some of the resources that we do give you as things that we are doing our best to keep as accurate as possible. I'm not saying that everything we say will always be the gospel truth, but what I will say is, again, you're seeing the scientific process in motion. So even if you are young, even if you are a child, elderly, everyone to this particular virus is vulnerable. There's levels of vulnerability, but everyone is vulnerable. So please do what you can to wear a mask, wash your hands. Please follow the CDC guidelines as much as you can so that we can all get out this epidemic together, because don't be that person that makes everybody hate group projects. Please don't be that person. The other thing I will say is I'm just very happy to not only have the support of the university, but also Janko as a chair. I mentioned that the initiative for the students that I had earlier, and I have to say that the department has been very supportive in kind of adding, helping administratively to support that particular program, so that we not only help our institution and help our society get better at fighting COVID, but we also help the next generation of scientists become scientists.
Thank you, Michael, for both comments. And I really want to particularly second the first one. I mean, it's hugely, hugely important to understand that this is, I lost a good friend, in peak health, a little younger than myself, to the disease, and those things happen all the time, unfortunately. So it's not a disease that's age restricted or restricted in any way, other than the fact that children are really, as a group, doing a whole lot better than the rest of us. And that's, I will answer all of the questions that you guys have posted in writing after this to the best of my ability. I would like to address one question that I picked up here, was going like, what does the university need to support its efforts? We live off of federal grants, and we write a bunch of them, and we get a lot of money in that way. The most difficult part is that those grants typically do not really pay for infrastructure. And so being able to have the right equipment, being able to have the right facilities, really relies on the university and the university's ability to build that. And right now that is very severely constrained due to the absence of the students and due to everything else that's depressed our economy overall. So support at this point is more critical than ever, and it would go exactly to those types that I have outlined, the infrastructure that enables us to do, like a new machine that helps us diagnose things in the lab, to image things, to, systems that connect us in terms of informatics and so forth. All of these things are difficult to finance otherwise, and really rely on central and unrestricted funds that would come into the university. So if you can help, it would be wonderful, and it would be super timely. It would be more important than ever. And I would really like to thank you for asking all the questions and being our audience today. It's been a privilege to talk to you. Thanks.
Yeah, thanks everyone. And a recording will be shared with all of the attendees, as well as the resources and the answers to questions that we weren't able to answer.